Imagine walking through a forest at twilight, the air cool and still, filled with the scent of damp earth and pine. As the last rays of sunlight fade, the woods around you begin to change. A soft, ethereal glow emanates from the undergrowth, not from fireflies or fungi, but from the fur of a creature moving silently through the shadows. A fox, its coat shimmering with a gentle, pulsating blue-green light, pauses and looks at you with intelligent eyes. It then vanishes into the darkness. This isn't a scene from a fantasy film. It's a glimpse into a future that is rapidly moving from the realm of science fiction into scientific fact. For centuries, we have shaped the world around us, domesticating animals and cultivating crops, but we were always working with the raw materials provided by nature. We were editors, not authors. Now, the story is changing. What if humanity could move beyond merely modifying existing species and instead design entirely new ones from the ground up? This is the dawn of a new and profound technological era, one that will challenge our definition of life and our role in the universe. We are on the cusp of becoming creators in the most literal sense. The power to write living beings into existence is no longer the exclusive domain of evolution or mythology. It is becoming a reality in our laboratories. This emerging field has a name that captures its ambition and its artistry: genome crafting. It is the intentional design and synthesis of complex organisms by precisely editing, combining, and writing genetic information. This is not just about changing a single trait, like the color of a flower or the size of a fruit. It is about building a creature from its most fundamental blueprint, its DNA, with a specific purpose in mind. We are learning to compose biological symphonies. In these compositions, genes are the individual notes. The resulting organism is the grand, living masterpiece. The journey we are about to embark on is one of immense possibility and profound peril. It will take us from the microscopic machinery that makes this all possible to the revival of colossal beasts that once roamed the frozen tundra. We will explore the design of purpose-built creatures intended to solve our most pressing problems, from pollution to climate change. But we will also confront the monumental ethical questions and ecological dangers that arise when you open Pandora's box. The power to create life is the ultimate power, and with it comes the ultimate responsibility. The story of genome crafting is not just a story about science and technology. It is the story of humanity's next great leap, a leap that will force us to decide what kind of gods we intend to be. The age of creation is upon us, and it will redefine our world, and ourselves, forever. How do you write the book of life? How do you edit a story written in a language billions of years old, composed of 4 simple letters that code for all the diversity and complexity we see around us? For decades, the idea was a distant dream, but the key was discovered in a place few would have expected: the ancient battleground between bacteria and the viruses that hunt them. Here, in the microscopic struggle for survival, nature had already forged the perfect tool. We call it CRISPR-Cas9, and it has revolutionized biology more than any other discovery in a century. Think of it as a biological word processor for DNA. It provides a way for scientists to find a specific sequence of genetic code, like using a search function, and then cut, copy, paste, or delete it with unprecedented precision. The system works with 2 key components. The first is a guide molecule called guide RNA, which is like a genetic bloodhound. Scientists can program it to hunt down and bind to a very specific, matching sequence of DNA within an organism's vast genome. The second component is an enzyme, a molecular scissor called Cas9. This enzyme is attached to the guide RNA, and once the guide finds its target, the Cas9 scissor makes a clean cut in the DNA strand. At this point, the cell's natural repair mechanisms kick in. Scientists can trick these mechanisms by providing a new piece of DNA. The cell then uses this new piece as a template, effectively pasting it into the location of the cut and rewriting the genetic code. This process, once incredibly difficult, time-consuming, and expensive, is now fast, cheap, and astonishingly accurate. It has given us a level of control over the living world that was once unimaginable. This isn't theoretical. The first generation of genetically edited organisms is already part of our world, serving as crucial proofs of concept. Consider the AquAdvantage salmon, created by inserting a growth hormone gene from a Chinook salmon and a genetic on switch from an ocean pout. This modification allows the salmon to grow to market size in just 18 months instead of 3 years, and it is the first genetically engineered animal approved for human consumption, a quiet harbinger of a food revolution. In laboratories around the world, another example glows in the dark. By taking the gene for green fluorescent protein, GFP, from a jellyfish and inserting it into the genomes of research animals, scientists can make specific cells light up under a UV lamp. This indispensable tool allows them to track biological processes in real time, watching how diseases develop or how drugs affect the body. But it is crucial to understand the difference between these early achievements and the grander ambition of genome crafting. The glowing mice and fast-growing salmon are the equivalent of correcting a typo or changing a single word in a book. They are products of simple genetic modification, altering 1 or 2 genes to achieve a desired outcome. Genome crafting is a far more monumental endeavor. It is not about changing a word. It is about writing entirely new paragraphs, chapters, or even entire volumes. It involves the complex orchestration of dozens or hundreds of genes, potentially taken from vastly different species across the tree of life, and weaving them together to create a novel organism with functions that nature never produced on its own. The tools we have today are the alphabet and grammar of this new language, and now we are beginning to learn how to write poetry with them. Before we can write entirely new stories, we must first learn to reread the old ones. Before we create what has never been, we must prove we can resurrect what has been lost. The first true test of our genome crafting abilities, therefore, lies not in the future, but deep in the past. It lies in the ambitious and awe-inspiring field of de-extinction, which offers a second chance against the finality of extinction. De-extinction is the bridge between simple genetic modification and true creation. A painstaking process of genetic reconstruction, using the genomes of an extinct species' closest living relatives as a template and then editing them, gene by gene, to bring a lost creature back into the world. The most iconic candidate for this biological renaissance is the woolly mammoth. For 10,000 years, these magnificent giants have been absent from our planet, their bodies locked in the Siberian permafrost. But their frozen tombs have preserved their DNA, allowing scientists to sequence their entire genome. A team of researchers, most notably at Harvard University, is now engaged in a colossal effort to bring a functional version of this Ice Age titan back to life. The process is a masterclass in precision genetics. They start with the genome of the mammoth's closest living cousin, the Asian elephant. Then, using CRISPR, they are systematically editing the elephant's DNA to reintroduce key mammoth traits. They are not just changing one gene, but dozens. They are editing in the code for the mammoth's shaggy, long hair, its thick layers of subcutaneous fat for insulation, its small, frostbite-resistant ears, and even a unique form of hemoglobin that allowed its blood to release oxygen in freezing temperatures. The goal is not to create a perfect replica, but a functional proxy. A cold-resistant elephant with all the key adaptations of a mammoth, which could be carried by an elephant surrogate or an artificial womb. The ambition behind this project is not just to create a biological curiosity for a zoo. The hope is that reintroducing herds of these massive grazers to the Arctic tundra could have a profound ecological benefit. By trampling down snow, they would allow cold air to penetrate the ground more deeply, preventing the permafrost from thawing. A thawing permafrost would release massive amounts of trapped methane, so the return of the mammoth could be a powerful tool in the fight against climate change. A similar story is unfolding for a creature that once blotted out the sun in North America, the passenger pigeon. Numbering in the billions, their flocks were so vast they were described as flying rivers. Yet, in a few short decades, relentless hunting drove them to complete extinction, with the last one, Martha, dying in a zoo in 1914. Now, the nonprofit organization Revive and Restore is using the genome of the passenger pigeon's closest living relative, the band-tailed pigeon, as a starting point. By identifying the genes that distinguish the passenger pigeon, they hope to edit the band-tailed pigeon's DNA to resurrect the lost species. Again, the goal is ecological, as the passenger pigeon was a keystone species whose disappearance fundamentally changed the forests of eastern North America, and bringing it back could help restore that lost balance. These de-extinction projects are our training ground. They are teaching us the art of complex, multi-gene manipulation. They are proving that we have the technical ability to perform large-scale genetic reconstructions. And once we have mastered the art of bringing back the dead, the next logical step will be to create the living. The Library of Life contains millions of volumes, each one a masterpiece of survival perfected by eons of trial and error. Evolution works without foresight, stumbling upon solutions through random mutation and natural selection. But what happens when a new author arrives? One with foresight and intention. What happens when we start to browse this vast library not just to read, but to combine chapters from different volumes to create something nature never intended, but the world desperately needs? This is the next great frontier of genome crafting, moving beyond revival and into purpose-built design, an era where we create organisms to solve our most intractable problems. Imagine our oceans, choking on a toxic soup of microplastics and industrial runoff, a problem for which conventional cleanup methods seem impossible. Now, imagine a new kind of creature, an envirobeast, designed specifically for this task. We know of bacteria that have evolved to eat plastic, but they are inefficient. What if we took the genes for those plastic-digesting enzymes and inserted them into the genome of a far more robust organism, like a species of fungus or mussel known for its ability to thrive in polluted waters? We could create vast colonies of these modified organisms, deploying them as living filtration systems in our rivers and estuaries to feed on our waste. We could similarly enhance the natural abilities of organisms that hyperaccumulate heavy metals like mercury and lead. By boosting the genes for specific proteins, we could create biological sponges, creatures that soak up toxic metals from the environment, which could then be safely harvested and removed. These would be ecological engineers, living machines designed to heal the wounds we have inflicted upon the planet. The applications extend from our environment to our cities, where we could create living infrastructure. Picture a city park at night, lit not by the harsh glare of electric lamps, but by the soft, living light of the trees themselves. A sustainable solution comes from glowing fungi, which have a complete genetic circuit that produces light as a byproduct of metabolism. By carefully integrating this entire set of genes into the genome of a tree, we could create living, self-powered streetlights that reduce our reliance on fossil fuels, cut down on light pollution, and reintegrate nature into our urban landscapes. Perhaps the most immediate impact will be felt in our food supply, as agriculture faces the challenge of feeding a growing population. Genome crafting offers powerful solutions, such as designing crops that not only resist drought but also fix their own nitrogen, eliminating the need for polluting fertilizers. We could engineer livestock to have a much lower methane output by modifying the genes that influence their gut microbiome. We could also edit genes to grant them complete immunity to devastating illnesses, improving animal welfare and securing our food supply. Each creation a living solution to a global problem. For every godlike power we acquire, a new set of demons is born. In our breathless rush to build a better, cleaner, more efficient world, are we accidentally designing its downfall? The promises of genome crafting are intoxicating, but the perils are equally profound. With every step we take into this new territory, we are walking deeper into a moral and ecological minefield. The power to create is also the power to destroy, and the line between the two is terrifyingly thin. This is the Pandora's box of our time, and we have already lifted the lid. One of the most significant ecological dangers comes from a concept known as a gene drive. In normal sexual reproduction, a gene has a 50% chance of being passed on to the next generation, but a gene drive is a genetic element that cheats this system, ensuring it is inherited by almost 100% of offspring. This allows a specific trait to be driven through an entire population with astonishing speed. Scientists have proposed using gene drives for benevolent purposes, like editing wild mosquito populations to make them incapable of carrying malaria, but the same technology could be catastrophic if misused. Imagine a genome-crafted organism, designed for a specific task, that escapes into the wild carrying a gene drive. Its engineered traits could spread like wildfire, permanently and irreversibly altering an entire species. Worse, if the engineered organism is more robust than its wild counterparts, it could outcompete them for resources, driving them to extinction and triggering a devastating ecological cascade. We have seen the damage that simple invasive species can cause. An engineered invasive species could be infinitely more destructive. Beyond the ecological risks lies a deep ethical maze. As we begin to design creatures for human utility, we must confront difficult questions about sentience, suffering, and moral status. If we create a creature with the capacity to feel pain specifically to grow organs for human transplant, what are our moral obligations to it? Is it a piece of biological machinery or a being with rights? Are we prepared to create a new class of sentient beings, born into a life of servitude, designed from birth to be used and discarded for our benefit? This looming crisis is compounded by a near-total lack of global consensus or effective regulation. The technology of genome editing is becoming more accessible every year, moving from elite university labs to smaller biotech startups and even amateur biohackers. Yet the laws governing its use are a confusing and contradictory patchwork that varies dramatically from country to country. This creates a dangerous situation where risky experiments can simply be moved to a country with laxer regulations. Without a robust international framework for governance and oversight, we are stumbling into the future blindfolded, hoping we don't walk off a cliff. Humanity has spent its entire existence as a character in the story of evolution, shaped by its indifferent forces. For millennia, we have been subject to the whims of natural selection, adapting to our environment or perishing. Now, for the first time in the 4-billion-year history of life on this planet, one of its characters is picking up the pen. We are transitioning from being products of evolution to becoming its architects. The future of life is no longer written solely in the slow process of mutation and survival, but also in our laboratories. Looking to the far future, the ultimate application of genome crafting may be in space. The dream of terraforming other worlds could be realized not by machines, but by the smallest of living things. Imagine sending legions of extremophilic bacteria and lichen to Mars, their genomes crafted to survive the planet's thin atmosphere and intense radiation. Designed to be hyper-efficient at photosynthesis, they would slowly consume carbon dioxide and release oxygen. Over centuries, these humble, engineered organisms would perform the first crucial step of preparing the red planet for more complex life. As we establish footholds on the Moon, Mars, and beyond, genome crafting could become the bedrock of off-world survival. Instead of shipping resources, we could bring a portfolio of engineered life. We could design living bioreactors, creatures that consume inorganic Martian dust and convert it into edible biomass. Others could secrete durable materials for building habitats or recycle waste with near-perfect efficiency. Space colonization may depend less on rockets and more on this new, applied biology, creating an ecosystem crafted by our own hands. We stand today at a watershed moment in the history of our species. The power of genome crafting is growing exponentially, and it will irrevocably change our relationship with the natural world. It positions humanity as a direct and intentional force of evolution. This is no longer about the slow, meandering path of survival of the fittest. It is about the swift, deliberate path of survival of the designed. We will be able to guide the evolutionary trajectory of life on this planet, and we will be responsible for the outcomes. This power is both terrifying and exhilarating. The power to heal our world or to break it, to create wonders or to unleash horrors. We are standing on the threshold of becoming the authors of the next chapter of life itself. And so, the single most important question we must ask ourselves is this. As we gain the power to write new chapters in the book of life, what story should we tell?